Okay, so yeah, this will be a seamless transition. So Anthony, when he approached me to come along and give a talk about my biology work, told me it was a semiotics meeting, and I thought, okay, that's great. I love semiotics. I know, it's nothing, but I'm interested in sign and representation and how things are, what we talk about, what they mean, and I think all of us are. So I'm going to talk about. He said, no, don't worry. You just have to talk about your biology research, and which was comforting because I feel like if I attempted to talk much about meaning, then I would be on thin ice. So I want to just tell you about what I do, and I've tried to draw out some connections between the things that I do in a way that maybe connects with the way you think, but it may not. So I apologize if it doesn't. So I'm going to first tell you a little bit about myself very briefly. I trained as a pure mathematician doing algebra, and so that's obviously very formal, very symbolic. And I came here quite a long time ago, and shortly after I came here, started meeting up with an old friend who's a biologist, and we started talking about epidemiology, so the spread of infectious disease. And after a few years of spending some very enjoyable time talking with Mark, we started writing papers. So it took me quite a few years to be able to feel comfortable enough with the biological language to start to work in it. And also, as a pure mathematician, I didn't really know how to do applied mathematics. So, it was quite an interesting transition. So I'm going to talk to you about the biology now, because that was what was asked. But I'm also going to try to draw some connecting threads. So I'm going to go through several different biological projects or paradigms, and they've got some commonalities, but they've got kind of slightly different questions behind them. So I'm going to just tell them to you, and then you can draw what conclusions you will. So the first one is epidemiology, so the spread of TB. This is where I started thinking about biology. So genotyping it means many things, but it's assigning some biological information or a type to a sample. So in this case, when people have tuberculosis, the diagnosis is often uses a sputum sample, so they scrape the inside of a cheek and they put it through some technology, and they say you've got this type or that type. And this is used by the epidemiologists to cluster the data, so that you can say all these people have the same genotype, so they're probably connected in epidemiological terms. So one of them was probably the source of the infection, and then the others were connected. So we wanted to know a little bit more about this, and we started to represent the information in graphs. So this is a graph which has each circle is a cluster of identical genotype, and the size of the circle is meant to indicate the number of people in that case. So the numbers in the circles, first labels of the genotypes, they're not important. And secondly, a list of numbers which are the sizes. This is data from 4 Caribbean islands. So the 4 numbers below there are the numbers from each island. So that's not important for today, but that's just to fill you in. So we represented the information using this simple picture, and using information about the way the molecular marker evolves. So we knew that this is spoligotypes, which is perhaps also not relevant. Spacer-oligonucleotide typing. They're just picking up bits of particular points along the genome, and they're saying whether there is a particular spacer present or absent in those spaces. So each one of these clusters has a particular type, and the arrows are connected if there is a possible mutation that takes you from one type to the other. So we're able to represent the data like this, and this tells you a little bit more than just saying, oh, if you're identical, then you're connected by transmission, perhaps. Because it tells you that if you've got directionality, so this cluster down here in this big one here has only out arrows, for instance. Okay, so presumably the source of the outbreak was of that type, perhaps. That might be an inference you might draw. So then these other clusters, there's 5 cases on one island here, the first island, and then there was a mutation in one of them. Now there's only one case of this type. For instance, so you can extract some useful information from this picture. But we found another way to find some even more useful information. Well, we thought it was more useful information, which is that there's something else that's different about some of these clusters. For instance, some of them have a large number, like this one, a large number of out arrows, a large number of mutations have been experienced by this type, and others have fewer, like this one, for instance, has only one. So there's actually information contained in that. So I'm going to show you a little animation. Well, this is a mathematical animation, which is to say it's not really an animation at all. It's a series of slides. So imagine you've got two strains, one of them spreading slow, one of them spreading fast, and you click the time counter. See that animation? So the fast one is growing after one time step a little bit faster than the slow one. The molecular marker that you're using to sort of monitor the cases, it mutates at a standard rate. So it's just ticking over its mutation process. It's a random process, but it's got the same rate everywhere. So over time, your fast one grows and you see the mutations emerging. But if you think about two clusters that are the same size, one fast, one slow, we're going to have to let the slow one catch up to get at the same size. So that means more time is passing in the slow one. I've stopped the fast one. So now I've got two clusters that are the same size, one of them which has been growing more slowly than the other. And the difference is that the mutation process of the molecular marker is giving you information about time. So you've got essentially flat data about the genotypes you're observing, and you can say that if you've got a cluster, for instance, two clusters the same size, one of which has experienced fewer mutation events, then it means that it's been around for shorter time, because the mutations are a sort of a clock, if you like, of time. So you can detect rapidly spreading clusters. So this might be kind of useful epidemiological information. So you might find a cluster that's spreading quickly in a particular environment, or you don't know why. This doesn't tell you why. It might be something intrinsic about the bacterium, or it might be something to do with the environment. It might be in a prison or somewhere like that where TB spreads very well. So I said in the title of the talk that everything is things and relations between things. So I'm going to refer to that sort of paradigm throughout. I know it's circular and it doesn't make any sense, but that's where my semiotics ends. So here the things are the clusters, and the relations are mutations that take one genotype to another genotype. So I've drawn a picture with things, which are these clusters of identical genotypes. So there's a particular genotype is the thing, perhaps, and then a mutation is the relation between the genotypes. So a second question relates to drug resistance, so more recently. So take that same information, you've got an outbreak of tuberculosis, you've genotyped a whole bunch of people, and you've also tested each one of them for drug resistance. So do they have TB that's drug resistant or not? And if so, maybe which drug is it resistant to, which drug not? So what can you say about that? Can you do something similar? Can you tell something about the rate of spread of things? So I'm going to show you a different kind of model, which is really quite different in this case. So it gets unreasonably complicated when you throw in phenotype and genotype. So phenotype is sort of an observed external property like drug resistance. So this model is very different. These objects are different. I've got a process here which is represented by this diagram. So instead of the representation of the data, I'm representing a process that gives rise to the data here. So I've got this shaded square is a set of untreated and drug-sensitive cases. So people who are sick with tuberculosis and not being treated, and the tuberculosis they have is sensitive to drugs. And down here I've got treated and sensitive. So there's an arrow going from untreated to treated labelled by tau, which indicates treatment. So you could be untreated and then you could become treated. So you move population. And this alpha going back is an infection parameter. It says, okay, you might have drug-sensitive tuberculosis and be treated, but in the meantime you might infect somebody else who doesn't know they're infected and so is not treated. So that alpha is saying that the people here might be causing additional cases up here. So we use this model. So this is what the biologists call a model. It's not what the mathematicians call a model, but it gives rise to a mathematical model, which is a bunch of differential equations usually, which I don't know how to work with, by the way. So we didn't use differential equations because, in fact, the equations are really complicated with this sort of model. It's too tricky. So we used simulations and a process called approximate Bayesian computation. So what we did, the paradigm of it, is you have some data, which is the outcome of this process, and you simulate the outbreak. So all of these parameters, I could explain all these other parameters, but maybe that's not completely important. I'll just run through them. The rho is a rate that a treated case becomes resistant. So we know sometimes that incorrect treatment or incomplete treatment can give rise to selection for resistant bacteria. So this is one of the ways that resistance is acquired by bacteria. And then if you're being treated and you're resistant, you might infect someone new who's now untreated and resistant, and then they might become treated again. Okay, so there's a bunch of parameters, and then you can be cured. And there's a rate of cure for drug-sensitive cases, which might be different from the drug-resistant cases. So you have a lot of computing power, and you run this simulation, and you generate sort of proxy outbreaks. And you take a sample from them to model what you might do in the real world, and you compare what you've sampled with the real-world data. And if what you've sampled is similar enough to the real-world data, you say, well, those parameters that I put in were similar, must be somehow representing something close to real world. So I keep those over here, and if they don't look like what I simulated, then I chuck them out and try again. And you do this 100,000 times or something. And the output is things like this. So these are distributions, posterior distributions of parameters, which just means distributions after the fact. So this one, for instance, these are 4 data sets, or 3, from Estonia, Venezuela, and Cuba, which all have TB problems. And this, for instance, is the relative fitness. It's showing that the fitness of the drug-resistant strains is about the same as that of the sensitive strains. If it was 1, they would be identical. So this is showing that it's consistent to say that the fitness of the sensitive and resistant strains is the same. So this was kind of relevant at the time we did this work, because there was a little debate that perhaps drug-resistant strains spread more slowly than sensitive strains, because by becoming resistant, they've become less fit. They've taken a hit by becoming resistant. They've survived, but maybe they don't spread as well. And that seemed to be the case in the lab. People could test it in the lab. And the resistant cases grew more slowly in the Petri dish. So this was kind of saying that in the wild, it seems to be not the case. And this one is showing that the proportion of resistant cases that are due to treatment failure. You could get a resistant case of TB 2 ways. One, you were treated and the treatment failed, and your TB became resistant. 2, somebody infected you with a resistant case. So this is saying that almost none of the resistant cases are due to treatment failure. Obviously some are, but a very small proportion. I think we found that over 95% of the cases were due to transmission, which is an obvious kind of public health corollary, which is to say, control your transmission. Okay, so that's epidemiology and a different kind of model. So what are the things now? The things now are sets of infected individuals. So they're not, there's a genotype going on under here, like in the first example, but here we've grouped things by phenotype, by drug resistance, rather than by their genotype. And the relations are movements. They're not possible mutations anymore, they're movements or creations. So the tau is a movement of an individual down from being untreated to treated, and the alpha is the creation of a new case. So this is more about the people than the bacteria. It's more about a sick individual. Okay, third example. So this is hopping around to very different examples of biological problems, so I hope that doesn't make you go to sleep or something. So this example is what is the distance between two bacterial species, and using genetic information. So bacterial genomes are circular, which is usually a surprise to people. It may not be for this audience, but the DNA is circular. So our DNA comes in 23 chromosomes, and they're all lines. Bacterial DNA has a single circular chromosome. And this is a little baby example with, say, 8 genes arranged around the genome, and two different genomes that we're comparing. And you can see that in the second one, a bunch of the genes have been inverted. A segment of the DNA has been flipped around. So the real numbers of genes are, of course, much more than 8. And usually we don't even talk about genes, we talk about regions of DNA that are preserved. But you can find sometimes a way to measure the distance between two genomes depending on the model of evolution. So the model means what you are prepared to allow to happen to these genomes that takes you from one to the other. The reason you might want to know how distant two species are apart is the same reason you might want to know whether we're more closely related to chimpanzees or monkeys. It's about understanding the history of the species. Sometimes in bacteria, of course, we don't even think of species because species are much more difficult to define for bacteria. So we just think two genomes, what's the distance? Can we maybe understand the relationships among these species, these genomes? So imagine you take the set or space of all possible genomes, of which there are many. Imagine you've got 60 regions or 60 genes on your genome. The number of possible genomes is then about a Google, about 10 to the 100. So it's a large space. And you're not going to compute with that space. You're not going to put a Google by Google matrix into MATLAB and play with it. So take this space in your imagination and put edges between the genomes if they're connected by a single event. So you're allowing a certain family of events to occur and you draw edges. And if you take some events like inversion, which is the example I showed where a segment is cut and reinserted with the opposite orientation, the set of inversions forms something very nice algebraically, which is a group. A group is a set with a single operation, multiplication say, and for which every element has an inverse, and there's an identity element. So it's a very simple algebraic structure, sort of thing you encounter in pure maths, second or third year undergraduate studies. So don't feel bad if you haven't heard about groups before. But groups are very beautiful symmetric objects. And you can represent a group, a finite group, by its Cayley graph, which is exactly the set of the elements and the edges between them if you can get from one to the other by multiplying by one of the particular set of generators in the group. So this has a really close analog with this biological problem. So this old Arthur Cayley, somebody should correct me, Michael, you know about Arthur Cayley. Can't ask the PhD students in the talk to give you the answers. But yeah, so about 100 years ago. So a lot of this material group theory is very old. But the biology is very new, obviously, because we didn't know about the DNA until very recently. So we can model this space of genomes with a very old mathematical object and ask those questions about it. What is the distance between two genomes? So there's this correspondence between the genome space and this graph, which comes from the groups. And your question about the distance between two genomes is the same thing as finding the length of a minimal path on a graph. So this particular graph is the symmetric group on 4 letters, which has got 24 elements. It's the number of rearrangements of 4 things. That's all it is. It's a very nice little example. And it doesn't look perfectly symmetric, but it is in fact symmetric if you had it in 3 dimensions. Let's see it. So the things here are now bacterial genome arrangements. They're not sets anymore. The last 2 examples I had sets. This time they're just genomes. And but the relations are sort of similar to the first example. They're possible mutation events between things. Last example, phylogenetics. Phylogenetics, already mentioned. This is a really proto-biological example, and that picture is seen in tattoos at phylogenetics conferences. I've got several friends who have various parts of this tattooed on their bodies. So this is from Darwin's notebooks in 1850s. I don't even know the date of the notebooks. But yeah, so he was musing about the relationship among these things that he was observing, and he wrote this, I think, which is a lovely, and that in particular, that I think has been the subject of many tattoos, which is not relevant to this talk. Anyway. So, yeah, I got sucked into phylogenetics because phylogenetics motivated some of that previous problem, the bacterial genomics problems. Because why do you want to know the distances between two bacterial genomes? Partly, you might want to reconstruct a phylogeny. And that might tell you about the history of the outbreak as well. So it might not just tell, you might not just care whether humans are closer to chimpanzees or llamas. You might care about how the outbreak has been going, and the information from the phylogeny might give you some of that. So, but one thing that really got me interested was that people have, in the last, actually, I think the first people talking, this is phylogenetic networks, the first people talking about phylogenetic networks might be a couple of decades ago, but it's exploded in the last decade or so. So these two pictures are phylogenetic networks, which is to say they're not trees anymore. So the top one that Darwin drew is an unrooted phylogenetic tree. It's a tree because it doesn't have any loops. You know, it's just the branches growing out like in a natural tree. But these are two examples below, and the one on the right is very difficult to see, I know. But these two pictures here are much more recent, and they're emblematic of a certain philosophical debate about deep phylogenetic history. So Ford Doolittle, in drawing this picture, was saying that life is fundamentally not tree-like. It's fundamentally network-like. Evolutionary history is too tangled up, especially at the base, to really say that there's a sort of a tree-like structure, because some people think it is tree-like. In particular, Eugene Koonin thinks it's tree-like. So that's just a couple of years ago. So this picture here, again, just in case you can't read it, this heading at the top here is bacteria, eukarya, and archaea. So so everything interesting with us, Animalia is this little branch here. So we're up there somewhere. So in the middle here, everything is kind of tree-like beyond there. It's not shown. Mostly, let's say it's mostly tree-like. But bacteria is, it's still completely not tree-like. There's horizontal gene transfer and there's hybridization happening all the time. So Doolittle's point here, to crudely summarize it, was that life is not very tree-like, and Koonin was saying actually it is, and he's got bacteria down here, and he's drawn this picture which is like a big tree but with some branches sort of going between some of the edges of the tree. So it's kind of a tree with some extra edges thrown in. So that was kind of an interesting thing to think about. So I'm not going to tell you much about any of these projects, just to list a couple of ideas. A collaborator and I tried to define what tree-like means, so formally, mathematically. So we defined a thing called Treebased Network, which is easy to test for, it turns out. And so this is an example of a wheat phylogenetic network published a couple of years ago, which is a phylogenetic network. There's bits of hybridization going on here, see? So in plants, you get a lot of hybridization happening. Thanks. Other things you can ask are, identifiability questions. So you might have some sequence data. You've got a bunch of sequences, genetic sequences, and you want to know, this is a sort of fundamental informational question, what tree or what network it came from. So that's a hard question. So if it came from a tree, it's possible to tell. If it didn't come from a tree, it's very difficult. So, and it depends on what model of evolution on the network you're prepared to live with. So we've worked a little bit on that. Consensus problems are problems about, you might have a set of trees which might explain your data, and you might want to summarize them in a single tree. You might get a set of trees by, for instance, sequencing you might get one tree by sequencing one gene and looking at how the genes on a bunch of species are related. One gene is related among a bunch of species. And then you might look at a second gene and a third gene, and you get a collection of trees, and you might ask, what's the species tree? What's happening at a species level? So you need a consensus tree. So that's a sort of philosophical question about whether such things exist and whether they're reliable, whether you can draw reliable inference about trees. So I'm not going to tell you anything more about those, except let's talk about the things. The things are the set of extant species or taxa or genomes. The things are the leaves, they're the things at the bottom. They're your objects that you care about. And your relations are now a little bit different. And I think of the whole tree or the whole network as a relation. And I don't know how to formalize that, but I think that's you can correct me if you have a better idea about this, but I think that's the natural way to interpret that. The network or the tree is the answer to the question, how are these things related? So if you tell me, if you give me two bacterial genomes and you say, how are those related? I'll draw an edge and I say, well, there's a sequence of mutations that goes from one to the other. But if you give me a set of species and you say, how are these related? The answer is the picture, if I can get it. So somehow there's a higher dimensional relation, maybe. It's there. So the commonalities are that all of these problems are sort of diagrammatic, and they've got objects, things, which could be any number of different things, and then they've got relations, which are ways you can compare the objects that you're looking at, and the relations might be very nice directional things, or they might be complicated graphs. And there is a mathematical theory of this. It's called category theory, which is kind of at the abstract end of pure mathematics, and I'm not going to tell you anything about that, but Don's grinning. A little interesting for me story was that after spending some years doing mathematical biology and the last 4 or 5 years doing phylogenetics, I remembered I had this picture which again, it's not rendered super well, but this is a picture from 1996 when I was doing my PhD. And this is a Hecker algebra problem. So Hecker algebras I'm not going to tell you anything about, but I was trying to work out if you could construct certain important elements. It was a basis for the center of the algebra, for what it's worth. And again, so this is my pictures, and the pictures involve, each column is a conjugacy class, and each one produces an element. And I drew this picture, which is the whole algebra, which is 120 dimensional, but it's S5, symmetric group on 5 letters. So I drew this, and I said, okay, if you start with this one, then you go up here, and the arrows do this and that. And I've since got reinterested in this problem, which I'd put aside. So I solved the problem in one sense for my thesis. I said, I could prove that you could always construct these things with a nice algorithm. But what I really wanted was a way to write down a closed form. I wanted a formula which said, you give me this one and I can just go, whoop, here it is. I don't have to turn a handle on an algorithm. I can just write down a nice expression. And that's why I drew this picture. But when you look at that and you put it next to Eugene Koonen's picture, it's kind of almost exactly the same upside down. And I think there's a lot of theory now about phylogenetic networks, and I would just be so cool to be able to go back to algebra and use some phylogenetic theory, because the algebra has to be really annoyed. But the other thing I should say about phylogenetics is it's really linguistics. So a lot of the early theorems in the early 70s were from linguistics papers. People were studying historical manuscripts, and they were studying how they evolved when people handwrote manuscripts, and they introduced errors by accident, and those errors propagated. And they used phylogenetics to tell, the original source, or to perhaps guess at the original source manuscript, which I think is a really nice, interesting example. And I've been talking to a guy called Robert Mailhammer, who maybe some of you know, about language evolution. So people like him are interested in how languages change, and it's possible maybe you can use phylogenetics for the same problem. So thanks.